name is William Coe. I'm a rising junior at Downtown Magnets High School, which is in downtown LA, and my field of engineering is biomedical engineering. Over the summer, I worked with Professor Ken Yu Shen and my Shine mentor, Dr. Yuta Ando, whom are both part of the Laboratory for Integrative Biosystems Engineering. So what does a biomedical engineer do? Biomedical engineers design solutions to problems in biology and healthcare by developing medical technology such as artificial organs and researching the biological systems present in humans and animals. So one recent problem in cancer/stem cell research is that during drug development, the microenvironment or the surrounding environment of cancer/stem cells is often not considered. This is a huge problem because the interactions between the cancer/stem cells and their microenvironment play a really important role in regulating cell growth/differentiation. To solve this problem, the mission of Dr. Shen's lab is to develop models of cancer and stem cell microenvironments. I utilize these models to find new targets for drug development and treatment. To make these models, Dr. Chen's lab utilizes a technique called microfabrication, which is a process of making devices that can manipulate substances at the micro scale. So you can see here at the bottom, here's an example of a microfabricated device designed to simulate hypoxia or a lack of oxygen in tumors. So over the summer, I worked on 3 projects relating to the utilization of microfabricated devices and learned how to extract data from them. So my first project was assessing the functionality of 2 different microfluidics devices designed to transport drugs to cells. So a common procedure in Dr. Shen's lab is to use water mixed with food dye to test if the devices are actually working. So you can see here in the control device, it's designed to take in 2 solutions, blue and yellow, and keep them separate. In contrast, the herringbone device is designed to take in 2 solutions, blue and yellow dye, and mix them together. So blue and yellow make green. So I used a software called ImageJ to compare how much the color changed between the start and the end of both of these devices. So basically these 2 graphs just show how much the color changed in terms of the percentage of red, green, blue values. And in the control device, you can see here there's a minimal difference in the percentage of red, green, blue values, and that showed that the colors did not mix in the control device. And in contrast, in the herringbone device, you can see that there's a 90% greater percentage of green at the end of the device versus the start. So that showed that the food dye did mix. So basically my analysis just showed that, yeah, my devices were functioning properly. So in my second project, I learned how to extract data from microfabricated devices. So these are images of breast cancer cells that were stained with a protein called GLUT1, and they were grown in my mentor's micro device platform that simulated hypoxia or a lack of oxygen in tumors. And you can see that there's an increased expression of GLUT1 in the hypoxic condition versus the normoxic condition or normal oxygen levels. To analyze these images, I wrote a program in MATLAB that can count the number of objects above a certain intensity level. I chose 165. And then from that, calculate the average intensity, the lowest intensity, and the highest intensity. You see here that the average intensity in the hypoxia condition is slightly higher, and that's because there's more GLUT1 expression in the hypoxia condition. And for my third project, I calculated the stiffness of gelatin utilizing Young's modulus. And biomaterials like gelatin are often used to mimic biological tissue in microfabricated devices. And Young's modulus is just a number that describes the stiffness of an object. So the higher the Young's modulus, the higher the stiffness. So I created cubes of gelatin of different concentrations and placed a weight on top of the gelatin, which is the stress in the formula. And to measure how much the height changed after I placed on the weight, which represents strain in the formula, and calculate a Young's modulus from there. So I could have gotten more accurate results if I used an instrument machine, which is designed to measure stiffness, but nevertheless, I found that Young's modulus increases as the concentration increases, meaning that the higher the concentration, the higher the stiffness of the gelatin. So special thank you to Dr. Shen for this opportunity to experience research. And thank you to my mentor, Dr. Yuta Ando, my lab partner, Yuban Lee, and Dr. Katie Mills and the rest of the Shine team for supporting me throughout my projects. Finally, thank you to my parents and my high school teachers, Dominic Lucido and Scott Portloff, for allowing me to pursue this opportunity.